Hey guys, my name is Didi Jimenez and today we're going to go over the overview of location and function of major brain regions. Let's go ahead and start. First, let's start with the basics. The brain is an organ composed of soft nervous tissue located in the skull of vertebrates, which functions as the coordinating center of the body. This organ first begins as bulges in the neural tube in an embryo that develops into three parts: the hindbrain, the midbrain, and the forebrain. This structure then is commemorated in the form of ventricles, that are cavities containing cerebral spinal fluid. More specifically, in the hindbrain is a fourth ventricle, which is continuous with the central canal of the spinal cord. A cavity in the forebrain becomes the third ventricle, which leads further forward into two lateral ventricles, one being in each cerebral hemisphere. Now, going into detail, let's look at the brainstem, located on the backside of the brain, on top of the spinal cord, that consists of the cerebellum, the pons, and the medulla oblongata. The brainstem's main function is to be a relay center for sensory and motor nerve impulses between the pons and the spinal cord, and the thalamus and the cerebral cortex, and aids in controlling our breathing, heartbeat, the diameter of our blood vessels, and our deliberate movement. The first part of the brainstem is the medulla, which is on the lower end of the brainstem. It houses the nerves that run between the spinal cord and the brain, and it also houses the nerves for organs of the chest and abdomen that are for movement of the shoulder and head, swallowing, salivation, taste, and hearing. The pons, which directly translates to bridge, is a literal bridge located between the lower brainstem and the midbrain. Its function is to relay sensations of touch from the spinal cord to the upper brain centers. It does this through nerve fibers. These are many face and head nerves that regulate facial expressions, salivation, taste, and eyeball movement. Together, the pons and the medulla control breathing and the body's sense of equilibrium. The last main focus of the brainstem is the cerebellum, which is Latin for little brain. It is located on the back side of the head, tucked underneath the cerebral cortex. Its function is to relay impulses for movement from the motor area of the cerebral cortex to the spinal cord, and finally to the designated muscle group. It receives impulses from muscles and joints, meaning it is a site for rerouting, in some cases refining the instructions for movement. It also controls movements of the body, balance, and coordination of voluntary movement. With all this being said, the cerebellum isn't the initiator, nor the link in a set of impulses, but rather it's a site for rerouting and refining of instructions when needed. Weirdly enough, it stores sequences of instructions for frequently performed movements and for skilled repetitive movements. This is due to the fact of the left and right hemispheres that connect nerve tracks from the spinal cord, where our same side of the body with opposite side of the hemisphere comes from. Nerve fibers in the cerebellum also play a role in reticular formation of widespread network of neurons. These nerve fibers, along with nerves in the thalamus, form the reticular activating system, which is how we maintain conscious. This formation aids in focusing and distractions, movement, transmitting and inhibiting sensations of pain, temperature, and touch. It is important because it works as a nervous filter for many stimuli that act on the system. Next is the core area of the brain, formerly known as the forebrain. It is also divided into three main parts: the cerebrum, the thalamus, and the limbic system. This is the anterior part of the brain, so we know it has to do with perception, movement, and the body's vital functions. Let's first talk about the thalamus, which is made up of two oval masses embedded in the cerebral hemisphere that are joined by a bridge. The mass houses nerve cells that sort information from sight, hearing, taste, and touch, and relay it to the cerebral cortex. This structure is located above the brain stem between the cerebral cortex and the midbrain, and functions to carry sensory information from the cerebrum and the limbic system. Moving on to the hypothalamus, which is located under the thalamus. Because of this location, it connects the nervous and endocrine system through the pituitary gland. It is a part that first detects crucial changes in the body and responds by stimulation. It stimulates smooth muscle and receives sensory impulses from the said area, aka it controls heart rate, passage of food, and the contraction of our bladder. This area of the brain also translates emotion into physical responses, houses neurons that monitor body temperature, blood flow, and is the control center for the stimuli that underlie eating and drinking, and plays a role in the induction of sleep. Let's go on to the pituitary and pineal gland. They both function with the hypothalamus. The pituitary gland responds to the hypothalamus with an array of hormones, which regulate activities of other glands. Some of them are the thyroid-stimulating hormone, adrenocorticotropic hormone, prolactin, and the sex hormones. With all this being said, it is important in growth and reproduction as well. The pineal gland, on the other hand, is known as the third eye because it is controlled by neurons with light sensitivity. Therefore, it interacts with the hypothalamus, and it also produces melatonin. Now would also be an adequate time to go over the limbic system that forms a loose circuit throughout the brain. It is located between the thalamus and cerebrum. It is an arc-shaped structure that functions in controlling responses like hunger, fear, thirst, sexual feelings, pain, pleasure, and anger, and even docility and affection. Part of the system is the hippocampus, which is vital for recently learned info or our short-term memory. The last part of the core area we are going to go over is the cerebrum, which is located on the outer region of the brain and is divided into two hemispheres that communicate through the corpus callosum. It functions in helping movement, speech, sensory processes, and it determines intelligence of a being. The biggest part of the brain is the cerebral cortex, which consists of gyri and grooves. The deepest groove extends from the back of the head to the front, therefore dividing the left and right hemispheres. The central sulcus that runs from the middle of the brain outward to both left and right, and the lateral sulcus, another left to right groove, somewhat lower on the hemisphere and toward the back of the head, further divide each hemisphere into four lobes: the frontal lobe, parietal, temporal, and occipital. There is also a fifth lobe, the insula, that is deep within the parietal and temporal lobes, which can't really be seen. Without a doubt, casual research is needed in medicine because it can identify targets for clinical intervention. In neuroscience, patients are tested through existing data. Those who already receive therapeutic TMS and DBS. This approach takes a certain region of the brain that is causally implicated in a symptom, and it's simulated in order to modify the severity of the symptom. This is taking on the fact that if damage to a particular circuit can cause a symptom, it is reasonable to hypothesize that stimulation of the same circuit may relieve that symptom. Brain lesions can inform researchers about treatment targeting through brain stimulation. This will help in determining if the network hubs are causally implicated in the behavior. Targeted stimulation could then be used and result in meeting the coherence criteria. Take a look at the picture where lesion network mapping happens, and it demonstrates that lesions caused from a decreased will to perform actions are connected to the anterior cingulate, the warm colors. Intracranial electrical stimulation sites overlap with this network, the green circle you see, and can induce a will to persevere unlike any other. Nearby IES sites, the white. Now looking at the right picture, LNM demonstrated that lesions that cause disorders of face perceptions are connected to right posterior fusiform gyrus. IES to the right posterior FC but not the right anterior FG or the left FG. Causes a distorted face perception. These findings illustrate how stimulation and lesion can be provided and give complementary info about the function of a neurotic hub. In this study, the researcher hypothesized that better DBS, deep brain stimulation, targets can be identified depending on the connectivity of effective transcranial magnetic stimulation targets and vice versa. TMS sites that relieve depression and DBS sites that modify depression and breach lesions that cause depression were connected in the study through common brain circuit. On the top left, we have the whole brain connectivity that was estimated using a normative connectome database. On the top right, for 461 incidental brain lesions, this map was used in comparison with depression severity and resulted in a map of the whole brain circuit connected to lesions that increased depression severity pictured on the bottom. This same procedure was done for 151 incidentally variable TMS sites and also gave a whole brain circuit map connected to TMS sites that improve depression, which is the middle graph. The colors on this map were inverted because TMS sites that improve depression were expected to be anti-correlated to lesion locations associated with lower depression severity. For the image labeled C, the same procedure was conducted for 101 DBS sites and resulted in a map of the whole brain circuit connected to DBS sites that modify depression. So all in all three maps were all very similar, more than expected. To conclude, the studies mentioned give us different techniques that resulted in different levels of insight, the main focus of them being the use of lesions to create stimulations in order to produce results. Although this is a great tactic, organization must be considered, and recognition of the level of causal interference that applies to a study is a must in order to form an appropriate conclusion. Our presentation focused on lesion and stimulation studies that consider the degree of coordinates between modalities. The given samples increase confidence in a causal relationship, which is variable to determine the effect of the treatment modality, but it doesn't clarify the effect of the targeted circuit. By using tools and approaches that are further along the causality continuum, we can come closer to translating brain maps into clinical treatment targets for neuropsychiatric disease. Well, we have reached the end of our presentation. Below you will find a list of our resources in case you want to look further into it. But that's it, guys. See you on the next one.